for multiple sequence alignment and blast, right? So if you done the phylogenetic tree analysis means you can easily infer whether the genes or samples, how much it is related or how much it is distanted, right? Differentiated. All the things you can get, and pictorially you can explain that. So for that, for phylogenetic tree construction, you can use so many methods are there. Distance based methods are available. Particularly, you can use UPGMA neighborhood joining methods. UPGMA is nothing but the arithmetic mean for joining, and here is neighborhood joining method. Okay, this is about the principle, so you can clearly understand these things. Blossom, HSP, and UPGMA neighborhood joining methods are how to do a matrix in the theory class. Hope you have an experiment in the theory class to fill the matrix, to take the sequences and fill the matrix for the blast. So by this way you can clearly understand the scoring and how to choose and what is the diagonal and what the horizontal arrows are indicating. So don't worry about this. If you have doubts, further doubt means you can just tell me. We will discuss in the discussion time. Okay. And the protocol is very easy. For multiple sequence alignment, you have to take top 10 blast resulted sequences. So why top 10? You can take top 20 also, depending upon that. But you may not use more than 25 sequences because it give the very congested phylogenetic tree. So that's difficult to identify and clarity of the result. Okay, affect the clarity of the result. So you can take top 10. Otherwise you can take all the identical sequences if it produce more than 90, or you can start with 100 and you can end with whatever the 80, 70, the cutoff range. That's up to you. Right? Because why I'm saying is sometimes you may get so many identical sequences. You can get identical sequences 20 or 25, 30 identical sequences within the percentage identity of 100 percentage. Sometimes we may not get 100%. We may get at 70 is the toppers more. Sometimes 70 or 60 may be the toppers one. By this way, you can order the sequence by using the e-value and the percentage identity. So for that, in those cases, you can take the top 10 sequences to analyze, right? So anyway, you need an, more than 2 to 3 sequences for this multiple sequence analysis. After that, you can go for the omega, Clustal omega through Google server. Then after the sequences, you may see that protein, nucleotide, and the RNA sequences in option in there in the Clustal omega page. Just click that respective sample and you can go for alignment. Okay. After that aligning, you can see that how it was aligned, and you can give the color coding, is that aligned or not, all those things. So you can create the phylogenetic tree also. On those days you can create a cladogram and a real tree also, and whatever the positions available or whatever the options available in the current version, you can use that, but you have to create phylogenetic tree and for discussion and interpret the alignment. Alignment means you have the results are already shown by the server. So in the screen you just copy, paste, or just download, or you can see that whether you can find any conserved regions which are in the same color, denoted by the same colors in that, and locate mutations or insertion deletions if they are meant. Otherwise you can leave it, and then analyze phylogenetic tree relationship. You have to um, copy and paste the phylogenetic tree and discuss that how many clades are there, how related is that with among the samples and among the genes or among the proteins, right? So repeat the same protocol by using your gene of interest. So previously the entire experiment will completed by using the protein, and here the same procedure you can use the gene of interest, right? So up to this, up to the eighth point, you can do by using the blast result, right? In blast, it will give the closely related sequences. So you may not get that much deviation or that much different spaces. right? So here, what you have to do is you can find the orthology sequences, right? You can do the same analysis for the orthology sequences and produce the phylogenetic and see how it is. Okay, and here you have already familiar with this thing. We have I have taught you in the first class itself, I think. So how to get the orthological sequence of a gene or a protein from the NCB, right? So you have done with a cobalt there, but now you are going to do with the same thing with the Clustal omega. That is the difference. That just do that and see how it is. Okay, shall we go to the practical session? So this is just go to the Google and type a Clustal omega and see that I will go here. Oh, before that we need a sequence, right? So protein, DNA, and RNA. And the protein I already show you in the blast, I think. This time I will go for DNA. So for I need a DNA sequence. For that I will go to blast. Then so blast. It's opening. I'm going to do with the nucleotide. So I just click here. Blast N default is selected. I'm going to choose the file. Right. I already have the file. Uh, in your case, you also had the file, I think, because you are working, keep on working in the protein and this thing, right? So you can go for sequence analysis now. And here, files, I have gene sequences here. If I want, I also have the protein sequences, right? So gene sequence of hemoglobin. So I give the hemoglobin here as a title. In that you can choose that standard databases of non-redundancy database. It means the errorless. There is no error in that. It will combine all the samples in all the available databases. So probably you can use, most probably you can choose this, right? Okay, then if you are working in the rRNA means, suppose if you are working in a project in which you are going to find the uh, phylogenetic, find the sequence, find the species, strains, means you can go for rRNA databases. See that 16S rRNA. Mostly we are using the 16S ribosomal sequences to identify the species or the strains, right? So you can go for that if you are really working in bacteria, not elements. Then you have the other option for the human genome proteins, also the mouse also. Then baculovirus experiment databases also is there. If you want to find any strains, means you can go for this prokaryotics and eukaryotics virus and others, right? Okay, for this this condition, I am going to use this nucleotide databases, non-redundant databases. It is not a special database. It will combine the results of all databases. So we can use NR here. I just leave all the things as default so that and just give the blast. So blast is running. It will take time to give the result. See that. In my case, what happens? See the personal identity, everything is 100. So by this way, how can I choose? I cannot give all the 100 sequences for the phylogenetic analysis. So I will take the top 10. Okay. So here the E value also 0, 0, 0, 0, 0. So I will go for top 10 sequences here. 1, 2, 3, 4, 5, and 6, 7, 8, 9, 10 sequences I have selected, right? So I need the faster format of the sequences. So I will go just click here. Don't go for individually, which means clicking here the action and get the sequences and aligning that in a separate file. Don't do that. You can get all the 10 sequences. Just select here, tick here, and go to GenBank. It will produce, right? For proteins, here you can find the NCBI link. Just select here and you can go for the NCBI link and you can get all the 10. See that I got it, right? So I got everything, all the 10. So here you can just click or select all the 10. So selected, and then you can go as usual, go to the file, and I need a faster format. So create a file. So file will be created. See that sequence faster file will be created. It may be in the download options. If you want to check, you can check that how many are there. Okay. So Control F. This is the faster symbol, right? Greater than symbol, faster format symbol. Every sequences will started with this symbol. So we can search that how many symbols are there in the entire file. This is the first one. 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10 is there, right? No, 9 is there. So I will check again. This may be the first one. So the first one, number one, number two sequence, third sequence, fourth sequence, and fifth sequence, sixth sequence, seventh sequence, eighth sequence, ninth sequence, 10. So only 9 is here. Shall I miss anything? Item selected. Oh my god. I see that there is not selected here. So I can select this also. Now I can create the file. Send to file, right? Faster format, create the file. That was for sequence 2 faster now. So now it contain all the 10 sequences. So now we can go for the blast. Sorry, go for the multiple sequence alignment, right? Clustal omega. I'm going to do with the DNA, which means nucleotides. So I can choose the file I have chosen from the downloads this sequence, right? So open sequence file is chosen. Then you can go for leave it as such, whatever here. If you want to give title, you can give, otherwise leave it and submit. So the sequence is submitted to multiple sequence analysis in the Clustal omega. So it will take time to get result. So after submitting, the result will get pop out. Okay. And while it is running, you can go for the orthologous sequence searching, right? Just you go here and select nucleotide, right? So just give the nucleotide or gene, P nucleotide, and we are working with the hemoglobin, right? HBB. So just I will put HBB here and Homo sapiens hemoglobin. I will get search. See that this is familiar for you. You can get the orthologous genes, right? For protein also you can get the identical protein option here or orthologous protein option here. So you can just here click here, you can get some results. Okay, we will see still the job is over in the Clustal omega. You can just go there and click view results, right? So the results will depending upon the network signal strength, you can get the result as much as fast. So here I have low connectivity. I think it will take time. In the meantime, I will go here and select the orthologous. Right? See that there are Homo sapiens, rat, macula, then other orangutan. So this much sequences are there. I need again. I need this architect's reference changes, right? So this much genes are here. I now select amino acid 147, 147, everything is 147. And the reference sequences is not available here. There is no option for reference sequences. So I will select all the 17, right? In previous experiment, he will go for you might go win for the protein alignment, right? And this I will download the faster format. Let me try to download. Notice what is happening in add to cart. No need download. See that download reference transcript faster format. One sequence per gene. So I will download this. So this is different file. Okay. And just we go here. See that in the Clustal omega results are shown. Tool output, right? Tool output results. In my case, you see that there is no 1 to 68 amino acid and let's say 61 to 128 amino acid. Likewise, it is going on. No one is get matched with that among the given samples, right? And what happens? The first match alignment is found at here, and this is the L2 647.1. We have to see that what sequence it is. It is bound with the 13 bases will get aligned with this at this stage. That what happens if you go further, you can see it is happening at 1980, which means what is that 1921? This is 1921, right? From the base of 1921, it gets aligned exactly. It is started here itself, right? You should see that what is the number of the base here. This is 1920, right? So 1920, 19 and here 18, 17, 16, 15 and 14 is the same, right? 1914, from the 1914th base everything get aligned. So this symbol shows 100% match, right? It's aligned. So like that, there are so many symbols are there. In my case, there is no other symbols has given is resulted, and star, colon, dot, hyphen, space are used. So you should know that what it denotes. For example, here stars are denoted for the 100% match. So see that everything is matched. So everything is matched. All the given sequence are matched. This is because we have given as a blast result, right? So everything is 100% identical. So it goes like this. Okay, it ended with this sequence. So you should mention up to which base the number of position of the base, starting and ending, right? So this maybe you have to count and discuss with it in a result, right? This is the original alignment results of the given samples. When if just come to the end of the result, you can find the option. See that alignment with the colors. Just click and you may find the colors of alignment, how it is, how many bases are in the same adjacent, like that. If you come to the alignment, you see that all the A's, all the T, adenines and T, these thing colors green and red shows it's aligned, how it gets aligned. Okay. So and you have to see this thing also, whether it is 100% aligned or not. So this is how you can find, see that how many adenines are there in that. Right? This may be useful to find out the domains in the protein or genes, right? So like that you can just download and or you can just uh, take a snapshot and paste as a result and discuss it what kind of confirmation you get. Okay, and next you can go for here alignment, right? We will wait that this will be show you the color coding into the color coding. Okay, see that 10 sequences are given, right? And you can zoom it. See that zooming happens. So nothing is aligned at this stage, is it? So zoom it and you can move that. So you can go for not I have selected something. So this is very big, so it contains I can zoom it from here to here. So I can't find anything in this. Okay, I will show you. I will back to this after some time. Okay. Now next you should go for the alignment and you should see that how it is aligned. This is okay. I have took a wrong experiment, wrong example I think because it will show everything. For that we should not select anything. Which one is given? Nothing is find here. Oh, what happened? I don't know. Okay, I will come back after a few minutes. Guide tree, then this is the guide tree of the phylogenetic tree analysis. You can get the phylogram here, phylogenetic tree here. It will take time, I think. So we will go here, phylogenetic tree, and find this. So it is getting late to the. Okay, see that. If you want to read it, you can go for read it. You close it, you can get it closed. If you want to expand, you can expand here. So it is not okay. So only numbers, everything is in a single origin. So that is the problem here when you are working in a blast, right? So everything is correlated. Everything is closely relative. So there only one root is available here. So all the clades will be in the single, I think, maybe. Okay, we will see in the next example. And here also see that here it is loaded, right? In the guide tree, you can see the phylogram. So here it will work, I think. Oh, to load. Because of the network issue, it doesn't show response. You can just expand and you can reduce that. You can see that. Okay. So what you have to do is you just say what is the length of that given length. Don't use the vertical axis. This is nothing but it is not going to use anyway. So you should see that what is the length of in the horizontal branch, right? So this is the root. Maybe there may be a common root, unrooted tree. This thing. So it has been and this is called as branch, and these are the clades. See, this many organisms are in the same. Are this many genes? Are this many proteins are closely related? Right. This is how you can create a tree. And the same results we will get here also, but I hope this is not responding. So hope you will get that. See that scales only get expanding, but it is not respond to me. Okay, leave it. But I hope you can get the result, right? Suppose if you want to see the results in a viewer means you can go here and you can send to simple phylogenetic. Same tree you can get it, right? That was sent to this and you can just use submit. We will see that what is coming. Okay, phylogenetic tree organization, how it is built. Okay, blast. Okay, then so still job is running. See that you can view the result. The same results you can view here. If you are using this option means you can change the node size, length, and algorithms. Okay, that's why we can use separate window. See that it take time to create. Same thing is coming. So maybe in that much close. Actually, all the options here should be loaded, right? If not loaded means there is a problem in the network, which means in the internet. I just try. Okay, let it be. I will come back after a few minutes whether it is working or not. Okay, this is how you can get a phylogenetic tree. Now come to the next one. Same in the Clustal omega, right? You can go for DNA, and this is for orthologous sequence, right? Then choose file and go for downloads. This is the orthologous transcript which you already downloaded, right? See that why it is delete this thing. Then to file was chosen and just go to submit. You will see identity tree is created or not. This much only created for this blast. So let me know how the results are for you, right? Which means all the samples are closely, very close relative. So I can produce, but here this option, if we click here, it will expand in the vertical axis, but it is moving in the horizontal axis now. Okay, if you want to see that everything is very close relative. That's why I didn't get the phylogenetic tree. I, I think. Okay, so leave it. And here, job is finished. For the orthologous to view result, I'm expecting this will be somewhat different to show you the results, which means we can access so many deviations because it is not that much close relative sequences, right? It is from different species also. So we will see what is coming as a result. Okay. So if you want to select here, you can select the only the based on the architects. Otherwise, you can select only or 5 or 10 spaces. Okay, that's up to you. So we see that the tool output came. So I have to see that. See, these are all the given orthologous sequences, right? You should remember that this is Homo sapiens, rats, and all those things. But here, what happens? They have given only the number, accession number. So see that there is star, star, there is a gap, there is a gap, there's a gap like that. And here stars and gaps only available. In some sequences you may get colons and semicolons, colons and hyphens also. You can go for alignment with the color. See that this is the alignment. Gaps are there. Okay. So this is not that much efficient also. And it means in the for the explanation, right? Suppose if we get something colon dots means I can explain you, but in the sample I didn't get that. Okay, that's how you can create. We will see what is happening in the phylogenetic tree. It will take time to create a tree, I think. So we will wait. The simple phylogeny, that's okay. We will close here, and I will close this also. We can see that this is the exact tree, right? Now you can expand vertically as much you want, right? So see that what is the branch, what is the leaf, what is the node, what is the clades, or that your job is you have to find what is it, what is the it source, right? Whether it is what kind of species it is, you have to name it and you can submit. Instead of giving the accession number, you can give species name itself in the file, right? The where is the problem arised is see that in the download option, if you open here, see that this is the number they have given nm and 1.0000518.5, right? So if I go here, where is that 51.8? Control F5 1. So what is the result? 518 is here, right? Is here. So this is respective for what is it? Homo sapiens, right? mRNA Homo sapiens. So instead of giving like that, you can just edit this. Edit that. Okay, it will start with the Homo sapiens, right? Subunit. Homo sapiens subunit. That's okay. And here also you can give just ratus. And here also you can delete. This is predicted. Mostly you can omit the predicted structures, right? That's why we have to select only the reference. Here also it is predicted, right? I have used without seeing this. In the real experiment, you can omit that predicted sequences, right? And here also you can delete. So there should not be a gap between the, I hope, right? Okay. Just so if you have found any predicted structure means you can omit that. You can take only the reference sequences. So there are 17 sequences we have given. So it will take time to delete everything. I hope I will do this is enough, right? So rest of the thing I will delete. For example, right? So I can delete. You can do slowly. You can complete all the editings. Now I will submit. Let's see. So we have deleted all the accession numbers. So what happens? It will give in the name. So just I will show you how it will work out. This is about result. Go for home again. That is the name. Choose file. Submit. Submitted. Wait for the result. Okay, I will show you another example. In between the gap, okay, results came. I have viewed the result. Waiting for the loading. So you can go for examples, right? So protein sequences. Uh, in the server page itself, they have given an option to use examples. I just I click you for the demonstration purpose. So just click. So they have given all the. They also take hemoglobin, but alpha hemoglobin of CA, free, and the hemoglobin of subunit alpha. See that there are next species name, bit number, and mouse. Right. This is how they have given as in an example. But when you are using this, use the example. I have clicked. It was loaded. and I just give the submit. See, this is our result. It is getting loaded. See that all the 17 sequences, as repeatedly they have given the NMX result. So that is failure. We cannot change anything, right? Gain is there. In phylogenetic tree. So again this number is coming. So that is failure then. Can you give this is the example sequences result. So here you can see that the different species. So we'll see how it is going. So alignment, guide tree is not still created. So it's created, but picture will not come. Okay. Then what about the tools output? It's taking no time. See that. Good night. See here, star, colon, and this colons are there. Gap is here, and dot is here, right? So your job is you have to find, right, what it denotes. I told you star means match, 100% match. Then colon means what? See that yes, yes is there, and here T is there. That is replaced by T. So you should find that and do as a homework. Otherwise, I will tell you in the next class. Right? Do as a homework what it denotes. Okay. Then there is no semicolon. Okay. This is how the alignment results will get uh, analyzed. Okay. So your job is you have to uh, find what it denotes. What is meant by star? What is meant by colon? What is meant by space? Sometimes hyphens may come. It means substitution, and in the substitution you can find polar amino acids with polar amino acids and the purines with the purines. So that substitution will be allowed sometimes. So what kind of substitution it is, then what kind of mutations happens. So everything you can get by using the symbol alone. So you go and read that. If you if you cannot find the description means you can just tell me. I will explain you in this class. Okay, and you can see with the score itself. This is the example sequences given by the Clustal omega itself, right? See here, clearly explained AKKS. And alignments, see that this is a color coding. You can see why this new sequences has been given. So it takes time to load. Okay, we'll see guide tree. It's not showing. It will produce a good tree, but here I don't have internet connection. I think here also you get all the sequences. Right, you can zoom it. You can find. See, it is loaded. This is how the sequence will be. Right. This is the entire sequence of our sample. If you zoom it, you can zoom and you can get that how the sequence. For example, they are given for the three sequences, right? For a multiple sequence alignment, see the M, all in the same cases. Here the M is given this color. So yes, here see that S is in green color, but here it is not matched with at least two. So the color gets very diminished. So like that, you can compare the result. If you give uh, 5 to 10 sequences, means all the 10 sequences will be lined up here and denoted by the colors. If it is, uh, it means if it is matched. Okay, this is how you can zoom it and you can paste it. Okay, take a snapshot of this thing for your result. You can paste it. So this is guided tree. Again, we have to wait for some time to load phylogram. Phylogen tree notations are given, but phylogram is not still loading. We have to wait for that. See that same thing. I am trying to remove this code, but it is coming. So we have to find alternative way to delete this thing, and instead of this, we have to put the what is it? We have to put that species name. We'll see what happens. Submission details. Resulted files are here. I think I didn't edit that. So if you edit it means you directly get the species name in that. You see that human, mouse, cat, and here it is not loading.